Our guest speaker today is Dr. Douglas Rosene from Boston University. Doug received a joint PhD degree from psychology and neurobiology from University of Rochester. He then came to Boston for postdoctoral fellowship in primate neuroanatomy at the Harvard Neurological Unit at Bethesda Memorial Hospital. He is currently a full professor in anatomy and neurobiology at BU. His research interests focus on the neurobiological basis of learning and memory in non-human primate models of dementia and age-related disorders, as well as neuroplasticity and functional recovery in non-human primate stroke models. Today he's going to show us some of his work on primate axonal imaging and tell us a little bit more about how it compares with diffusion MRI and tractography. So it's all yours.
Thanks. Okay. Well, I appreciate the opportunity to come here and talk to you folks. I've had long-standing collaborations with Nikos Makris here, Van Wedeen, Jeremy Schmamond, and I've been in and out of this building for years at 10 o'clock at night with rhesus monkeys scanning them up until about 5 years ago when we got our own 3T scanner. But it's always fun to come and talk science. So I am going to talk about the neuroanatomical and cellular correlates of MRI tractography. And the question as a work in progress is, what are we really seeing? So I'm going to show you mostly tissue-based studies in rhesus monkey brains. And those brains come over here for ex vivo scanning. George Dye and Van Wedeen are doing that. And in fact, we have a brain over here right now. George is out, but we're trying to connect to get it back so that we can process the tissue. So that's what I'm going to talk about. And all of the results, of course, were done by other people in the lab. Farzad Mortazavi, Senior Research Associate, and a brilliant immunohistochemist is sitting right here, and I may direct some questions to him if they come up. And Adrian Oblak, who left for a position at in the Alzheimer's Center at the University of Indiana, did a lot of the tract tracing and is still collaborating on this. So, again, the studies have been done largely in collaboration with Van Wedeen and Jeremy Schmamond, and I'll acknowledge them and the funding up front from NIA, National Institute of Aging, NIMH, and NSF for different aspects of this work. So a brief overview of connectional neuroanatomy, because what I'm going to talk about, depends on understanding that. So there are a lot of ways to look at how the brain is wired. Post-mortem brain dissection was, of course, one of the first things done hundreds of years ago. Myelin stains, when we could first cut fixed brain and process it, and one could look for areas of demyelination if there were a lesion and infer that a tract was running through that area. Neurophysiology, you can stimulate the brain and record in places, and short latency, apparent monosynaptic potentials tell you about connectivity. And of course, tract tracing originally grew up in the late 40s and 50s here in Boston with Wally Nauta and colleagues at MIT, looking at degenerating axons and axon terminals. So now, instead of just looking at the demyelination, you could actually see where things terminated. But again, you had to have a lesion. And it wasn't amenable to the human brain because it only worked with about 4 or 5 days of survival after the injury. In more recent times, tract tracing using the transport of labeled molecules has been the favored approach, and that's what I'm going to show you some examples of today. And then, of course, diffusion MRI imaging with tract reconstruction, first with DTI, and now more recently with diffusion spectrum imaging developed here. So let's look at a couple of fun examples of this. This shows subcortical white matter as it is dissected. A beautiful book by Glebevich has a whole bunch of these, and if you freeze and thaw properly and do dissections, you can see fiber bundles here crossing the corpus callosum, or here the geniculocalcarine tract from the lateral geniculate back to visual cortex. But again, there's some limitation on what you can see here. Myelin stains confirm exactly where the white matter is, but they tend to be extremely high contrast, like the Weil stain, where you can barely see individual fibers, although we've used a stain called black gold, which shows small bundles and can be titrated a lot better. Nevertheless, it's still difficult to know what's going on. Here's an example of how you can look at connectivity using electrophysiology. And Jeremy Schmaman, with my mentor Deepak Pandya, put together a beautiful analysis from some tract tracing data of the big bundles in the brain and where they go, and labeled them. But this is done with autoradiography, and while you can see the big bundles, you can't follow individual axons at all. And then here is modern from Van Woodeen in 2008, diffusion spectrum imaging, showing you in an intact brain how some of these fibers may run. And the question is, how does this correspond with the reality that we can see in fibers? So some problems that you're all aware of, the fiber reconstructions in MRI tractography depend on the resolution you have and on the algorithms that are used to connect voxel to voxel the presumed tracts running from one to another. So how can you validate these? Well, we can do microscopy and post-mortem tract tracing a little bit in human brains with dye, but in monkeys we can do transported tract tracing where you're depending upon physiological processes. So our approach for all of the histology that I'm going to show you is, first, the animal gets a pre-mortem T1 and DSI scan at 3 Tesla. Then in vivo injections of dextranamine, an anterograde tracer, are done in the animal in a neurosurgical procedure. We visualize or find stereotactically the area that we want to show. The tracers we've used are biotinylated dextranamine, and I'll show you pictures of that either chromogenic, where it appears black, or with fluorescence, where it can appear any color that we want to label it. And there are also dextranamines that come pre-conjugated to colors, fluororuby and fluoroemerald, that fluoresce red or green. Then the animal, after an appropriate survival time, is perfused, the brain's taken out, and it comes over here for post-mortem T1 and DSI scans at 4.7 Tesla or higher, usually over a whole weekend. And then we get the brain back and we process it for microscopy, where we can see individual fibers. So we can visualize the tract tracers using light or 3D confocal fluorescence microscopy, and we can also visualize the total forebrain fiber population with immunohistochemical procedures that identify individual axons. We're not looking at myelin, although that's a capacity that we could do with immunohistochemistry. So, to give you just a little bit of interesting history, here is Camilo Golgi. You can just barely see him here, sitting at his laboratory desk with all of these bottles of chemicals that the lab safety officer would just go nuts over if they walked into his place right now. But this really started modern neuroscience, because he developed the Golgi stain, which it turns out his colleague, not collaborator, competitor, Santiago Ramon y Cajal, applied with exquisite skill and microscopy to all kinds of tissue. And it's obligatory for all of us who think of ourselves as neuroanatomy to show that almost anything we're going to show you, Cajal showed over 100 years ago. So here is, in fact, a Golgi stain. This is a pyramidal neuron in the cortex, 2 of them with apical dendrites heading up toward the peel surface that would be way up here. And here you can see an axon coming off. You can tell it's an axon because it develops a uniform caliber, runs fairly straight, and has branches at right angles. That's been known for over 100 years, right angle branching in the cortex, but it turns out this in fact is a general principle that I'll show you about. So in fact, Van saw this using his analysis of the diffusion spectrum imaging in the cortex. This is a ex vivo monkey brain, and if you look at this area right here, you see that in the subcortical white matter, callosal fibers and fibers of the SLF, superior longitudinal fasciculus, or cingulum bundle are in fact crossing at right angles. It's not a bowl of spaghetti, as one of the popularized versions of reports on this study cited. So here, in fact, is one of the pictures from the science paper that was published a year ago in March, and Farzad and I were honored to participate with Van in. And this is the sagittal stratum, back in the occipital lobe of the brain, callosal fibers coming in, and cortical fibers out here superficially. More deeply, you would find geniculocalcarine tract fibers joining it. And when you look closely at that area, you find that it's in fact forming these sheets of fibers that cross at roughly right angles. So initial reviews of this paper suggested that people were going to have a hard time believing this. So Farzad and I took a monkey brain, ex vivo, perfused with fixative, found where the sagittal stratum would be just outside of the geniculocalcarine tract that stains a little more darkly here because it's so dense, dissected it into a couple of blocks that we could cut at specific angles to try to show these sheets, and what we saw, or what Farzad saw, was this. Now let me explain it for 1 second. You're seeing not myelin, but you're seeing individual axons. They're stained with an antibody called SMI312, commercially available. It's a pan-axonal antibody to neurofilaments. And so you have big fibers, big axons, small ones, and very tiny ones, and you can see that they're crossing at roughly right angles. And this is one 30 micron thick section. You go down deeper in it, and you see a similar pattern, slightly different angles, and in fact there seems to be, and we don't understand this at all, some space between the sheets that are formed by these. So here is a movie that Farzad made of fibers, okay, in 3 dimensions. So the XY is fairly large, but the Z here is only 30 microns. You can tell that these axons are all on the order of 1 to maybe 4 or 5 microns in size. Remember, it's not myelin that we're seeing here. The space between them is probably largely occupied by myelin and by, of course, the glial cells that are making the myelin and astrocytes that fill in the extra space. So indeed, this brain is put together in these sheets. Now, this is showing all of the axons in a particular area. Doesn't tell us much about how things are connected, but we can use tract tracers to learn a little bit more about that. So here's a rhesus monkey hemisphere. Here's the central sulcus, principal sulcus, arcuate. So this is the frontal lobe. Here's the occipital lobe back here. And what we did in this particular monkey is we put an injection into area 4, primary motor cortex, happens to be the hand representation, of biotinylated dextranamine. We put one of the conjugated fluorescent versions, emerald green, fluoroemerald, into prefrontal, area 9, into parietal lobe, area PGOPT, we put fluororuby, and we put lucifer yellow into area 17. I'll show you mainly sections through the injection site and around the area 4 motor cortex injection. I'll show you an example here or there of these others as well. Now, I think it's important to understand how these tracers get into the axons and go to wherever that axon is going, including all of its branches. And this happens through axonal transport. All neurons have a microtubule system that links the cell body to their axon terminals. And it has bidirectional, fast and slow rates of axoplasmic flow, and there are in fact little motors. In the anterograde direction, away from the cell body and toward the terminal, sometimes called orthograde, are little kinesin motors that carry everything from mitochondria to polypeptides and amino acids out to the terminal, neurotransmitters if synthesized in the cell body. There is another set of motors, dyneins, that in fact carry things in a retrograde fashion. In part, they're recycling everything that came out anterograde, but the terminal also tends to take up by phagocytosis and or receptor-mediated endocytosis all kinds of molecules, and those are carried back to the cell body. So if you inject something at the terminal that's a retrograde tracer, you find all the cell bodies that project to that area. If you inject something around the cell body that's an anterograde tracer, it goes out the axon and all the way into the terminals. And you can see all of this with tract tracing methods. So what we've been using mainly so far are the biotinylated dextranamines. They are taken up by dendrites and neuronal cell bodies, that's clear, and transported out processes. Why they're taken up is unclear. No one understands the mechanism. If you have a 10 kilodalton size dextranamine, it goes mainly anterograde. But if you want to see retrograde, you can use a 3 kilodalton dextranamine, and we've mainly used the anterograde so far. You can inject these in volumes as small as 1/10 of a microliter using fine point Hamilton syringes, although some people in mice or rats will use ionophoresis to get even smaller volumes. But this will label an area about 1/10 of a cubic millimeter in size. If you put in fact, 3/10 or the decimal point's in the wrong place. My apologies. That should have been 0.1. If you put in 2 or 3 or 4/10, you can get up to a cubic millimeter, but not much bigger than that. It's stable in the brain for 4 weeks, and this happens to be transported at the slower of the fast axonal rates, about 15 to 20 millimeters per week. The monkey brain is 65 millimeters long, so in 3 to 4 weeks we can have it go everywhere. And we usually use a 3-week survival time. Now, once the brain's taken out, how are we going to see that? Well, the brain is cryoprotected and frozen. We then fix it to the stage of a freezing microtome. This is out at room temperature. This little white thing here is in fact a frozen block of brain surrounded with dry ice. It's maintained at 50 degrees centigrade. This is actually a little well of alcohol with dry ice in it. And then the microtome can cut 30, 60, or 90 micron thick sections that thaw immediately onto the blade, and you pick them up with a paintbrush and put them into a buffer solution to save them. You can distribute them. We normally do every 10th, so that means we have a series that's about 300 microns apart. So from what we have about 3 sections per millimeter. But we can do every section if we want absolutely adjacent, or we can space them at 60 or 120, any multiple of 30 that we want, depending on the level of resolution. So here, in fact, is the face just after a section was cut. seen from a camera that we have over the microtome that photographs the face as it's cut. And the way the microtome works, the blade is stable, so the cutting surface is always in the same place, and the tissue block moves up 30 microns or multiples thereof every time we cut a section. So this is a little movie just to show you exactly how it's done. This is the hand of one of the technicians, or it might be mine. No, I think it was one of the technicians because I took the movie. And you can see how simple it is. You cut the section, it's coming up onto the blade. Don't go too fast or it cracks. Once you're off of it, here it is, and you'd reach over, take it off with a paintbrush, put it in a vial, and we know exactly where that came from because we've got a photograph of it. Now, for biotinylated dextranamine, the biotin will bind with avidin that has some kind of marker on it. Could be a fluorophore, but if you use the avidin peroxidase complex, you can deposit just by adding H2O2, diaminobenzidine, and nickel. You can deposit a nickel dab reaction product every place there was a BDA molecule. And when you do that, you get this kind of picture. So this is from that exact brain that I showed you. This is the BDA injection into the hand area of motor cortex. This is the superior precentral dimple, so the foot area would be up here. The face area down here, we're looking into the insula of the Sylvian fissure. Now, the very black reaction product here is, of course, the BDA where it was injected, quite dark. But those of you up front at least can see there's fibers streaming out here and forming a bundle. Excuse me, this is heading into the corpus callosum. But other fibers are going straight down here, and other fibers heading out this direction. How did they get there? Well, some of you who looked at Van's paper in Science may have noticed that shortly thereafter, Dr. Catani published an objection saying fibers don't do that. And here was his view of how fibers leave motor cortex. They come out and they make nice, graceful turns to get into all of these different directions. And that's in fact what's been surmised for 100 years from studies of tracers, with the notable exception of Wendell Krieg and a few others who looked very closely at degenerating axons and showed what looked like right angle turns. So we in fact have these individual fibers labeled where we can look at them under the microscope down to the level of a single axon. And I'm going to show you in the next couple slides this area where the fibers come out, and you can see already here that somehow, while there are certainly fibers that are coming straight out from the injection site to go transversely into the callosum, we already have fibers running at right angles to them. And I'll show you at a higher magnification these fibers within there. And here, in fact, is what we see. I've just labeled here with this key fibers that are making 90-degree turns. See one right here. Fibers, of course, that are crossing, there are plenty of those. We can resolve them by focusing up and down. And fibers that are making a T, the Cajal type branch, where axons branch at right angles. And remember, we're not seeing myelin here. We're seeing the axoplasm as labeled with transported biotinylated dextranamine. So let's look at a couple of higher magnification examples of this. So here's one of those T's, and you can in fact see this branch right here. This heads a little deeper, that's heading, and they're staying in plane. All right? Here's an example of one of those right angle turns. Now you might say, oh, well, look at that. It's really not a right angle, and it's not unless it's a branch, but it's over a very small radius, probably a radius of 3 or 4 microns. Not something that would be resolved in an MRI scanner with voxel sizes of, at best, 100 microns and most often approaching a millimeter. And here's an example of a turn where the fiber is turning up and out, leaving the plane of section. Okay? This was going to head out this direction, but the face of the section cut it off. And we can find these in all kinds of places. Here are examples that Farzad imaged using a fluorophore to make the biotinylated dextran amine fluoresce so that you can go into the confocal and take 1 micron or half micron stacks through the entire thing, and you can find anywhere you look fibers that are making right angle turns. Now wait, that's not right angle. Well, remember, if you look at something like this, it's right angle, but anytime it's not exactly in plane, it will not appear to be at exactly right angles. But they're very close. Here's another movie to show you a fiber branching. All movies, courtesy of Farzad Mortazavi. I haven't a clue how to do this. So you can see the fiber, okay, right here. Comes around. The T branch is right there. We can look at it from either side. Okay. And why do they look a little flattened? We get shrinkage in the z-axis, and of course the fluorescent scope always has this donut hole on the deep side of where you're looking. So, but this is a perfectly good example of how these fibers move. So now we have labeled fibers. What about looking at the total population of fibers in this same animal? So here's the same region where I just showed you labeled fibers coming out like this, going down like that, the centrum semio-valley deep to layer 4. And you can see that most of the fibers are at right angles, although you get an occasional one that's at an odd angle. Well, this is the same image, but processed by our collaborators, Gene Stanley and his graduate student, Will Morrison, in the Department of Physics, to identify the orientations and to color code them. So here, in fact, you can see fibers in green that have one orientation, fibers in sort of magenta blue with another orientation, and an occasional fiber here in yellow that's not adhering to either orientation, the outliers, if you will. And this offers the possibility of being able to quantify the total fiber population. And in this same image, using a different fluorophore, we can find within it the BDA labeled fibers and the orientations that they're in, and then of course the turns that they make. Now, is this organization of white matter simply a property of the sagittal stratum, a unique place, and maybe centrum semio-valley right under motor cortex? And the answer is no. Wherever we look, you find this. So we took out the cingulate gyrus right here. Here's the corpus callosum, and the cingulum bundle, as most of you know, or the cingulate bundle, as JCN made us call it in a recent article, not liking the term cingulum, runs deep to this. And here, in fact, is a SMI 312 immunohistochemical label of all the fibers reconstructed from the confocal in the cingulum bundle in this medial piece. I'll show you in a little while what it looks like caudally and rostrally. But Farzad used image J and did an analysis of fiber angles. And you can see here, this is 0, right around here. And here's a peak on the fibers at around 35, and there's another one out here at around 60, and the difference between the two is approximately 90. And he then did a 3D Fourier analysis to find the principal directions, and indeed shows this kind of orientation. And in the cingulum bundle, it's almost identical to what Van did looking at the sagittal stratum in the science paper. And this is reproduced from that. So again, it's not just a unique thing. Every place we look, we seem to see this. Now, do fibers turn at right angles within that? Here's a 3D movie that Farzad made of the cingulum bundle, and you might want to take a peek at. There's a fiber right in the center that we're going to zoom in on. Yeah, it's on the other side, that in fact makes a sharp right angle turn, and there it is, right there, moving out of plane. And you can see fibers in opposite planes. So, if we look at all of the cingulum bundle, here's what we see. Most of you know that the cingulum bundle has fibers entering it and leaving it at different points. It's not a continuous fiber tract. It is a multi-connectional fiber tract. And so you in fact find different characteristics of the fibers. Posteriorly, as they're going to go back around the splenium, in the medial section or in the rostral section. In all of them, the fibers that do cross through are crossing at right angles, and fibers that turn are turning at right angles to either leave or enter. We of course can't tell until we put tract tracers together. Now I'll show you one other only slightly related thing, just to show you a little bit about the capabilities that doing immunohistochemistry and neuroanatomical histology can add. This is a confocal reslice of an axon that was double stained. SMI 312 labeled the axon in green, and you can see a few little places where I hope it looks mainly green. And we used another antibody, EP antibody, that stains damaged myelin, because in our aging monkeys, we're interested in damage that accumulates with age. And so you can see the EP alone in the myelin is red, but where it overlaps with the green axon is in fact showing up as yellow. And we can of course do this with myelin basic protein as well, to answer questions about specific size myelinated fibers as we go forward. Few more examples here. This is a 3D reconstruction of the cortex from the pia down to the white matter that has BDA labeled fibers plus axons labeled with SMI 312. So here we have both of them together. And if we look down here at the white matter just beneath layer 6, you can see here green, a BDA labeled fiber that in fact is making a right angle turn. We'd have to have a little more resolution to be sure if that was a T or not, but it looks like it. And there are a few other examples of BDA fibers running within a group of red fibers labeled with SMI 312 that are mainly coming at you, okay? And here's just the BDA fibers alone at a higher magnification, again showing you examples of these turns. Well, how about another place to look? In the same brain that's been imaged, here's the superior temporal sulcus. So this is the superior temporal gyrus, auditory association cortex. And if we look in white matter beneath it, we get the same kind of crossing pattern, although again on this one, most of the fibers are coming at you in this particular section. But looking at gray matter produces a somewhat different picture that's a little bit more confused, and we're not quite sure what to make of this yet. But you can see here that with SMI 312, it's a little bit more like a bowl of spaghetti cut through there. We need to adjust angles and try to get complete 3D reconstructions in the gray matter to see what's going on. But Van has seen interesting DSI images of gray matter as well. So that's a sort of future direction. Now, how can we take the labeling that we see in one of these sections after we've cut it and get a picture of what's going on in the whole brain? And this is another work in progress. Here we have a brain that's on the freezing stage, and gee, this dry ice looks a little bit red. No, it wasn't a bloody perfusion. We added paprika to the dry ice so that it would be red, and it would help us to segment it out so that when we take a photograph of this block face, undistorted, just before it's cut, we can then extract an image without any of this around it. And we do that right now every 150 microns through the brain. So here's that block face image, and here's the section after it's been processed. You can see we've got distortion. The hippocampus and the temporal lobes have floated away. The sulci, are opened up a little bit more than they should be. A goal in the long run is to use these images when the section gets mounted, and it gets mounted by one of the people in the lab with a petri dish, a microscope slide, and a little paintbrush floating it up onto it. And you can use these pictures, if you print them out, to make sure that you put everything back together the way it should be. This has been stained with neutral red. That's why the gray matter looks a little pink. And the biotinylated dextranamine is stained blue-black with the nickel dab, and here you can see some termination patterns. There's a projection pattern in the thalamus. There's another one down here in the subthalamic nucleus. And so we have all of these tracers, both the axons and the terminals, in these histological sections that can be adjusted to correspond to the undistorted original frozen section. So what can we do with this? Well, here's the nickel dab label, and here's the nickel dab label after we do a color segmentation, get rid of all the red and the white, leave only the blue-black, and then project that onto the image of the corresponding section. And here's the tracer, and some of you up front can see these little black lines. Here are the fibers as they're leaving the injection site and entering into the grid. Well, what we want to do is take these pictures, extract each one 150 microns apart, process every section 150 microns apart, and then project them onto the undistorted image, correcting for any kind of distortions that occur, like right here, the third ventricle tore open. That's an image processing tour de force, but not impossible. Where are those sections going to go? Well, they're going to go into the 3D volume here that Adrian Oblak reconstructed from the photographs of the block face. So we have a photograph of every section, and we now have a camera back that will let us photograph at high resolution the BDA, or immunohistochemically processed section, line them up, and then we have a true 3D volume here that will have within it, the labeled fibers, and can be warped to, and without much distortion, because this is the frozen brain before any histological changes, warped to the DSI images of that very brain. So we'll be able to follow where these tracts go, where they turn, and what's going on there. I'll just finish up, show you a couple other kinds of things that can be done with immunohistochemistry in gray matter. Here, in fact, is immunohistochemistry for SMI312 again, the marker for axons, and in red, NuN, a marker for neuronal nuclei and soma, although it's mainly in the nucleus, a little bit into the soma. This is a different combination where we have green for NuN for neurons and blue MAP2, microtubule associated protein that labels mainly dendrites. And so a purely green cell is one that has little MAP2 in it. A bright white cell is a soma that had both MAP2 and NuN, and the dendrites are purely blue. So we can extract these different features because all of these would in fact restrict diffusion MRI signals and will let us look at the gray matter using this fantastic microscope of diffusion MRI. And so here's one last example, the junction of the gray and white matter with MAP2, SMI312 in red for axons and green, NuN for neurons. And here at a higher power, you can see, except for the glial cells, we've got almost everything that's here in gray matter. It can be extracted with color separation and analyzed in all kinds of quantitative ways. So, conclusions. What have we learned so far? And as I said at the beginning, this is a work in progress. But what we seem to be able to say definitively is that immunohistochemical identification of white matter axons confirms the predominance of grid structure, right angle sheets within the white matter of the monkey brain. And we want to apply some of this to human brain sections and have been talking with Bruce Fischel and Gene Augustinac about doing that in the near future. With our injections of tract tracers that label specific fiber populations, we've demonstrated that cortical tracts conform to the grid. As those fibers leave motor cortex, they're going to be in one or another of those orthogonal patterns almost as soon as they get below the gray-white matter junction. And the microscopic assessment of these tract tracer labeled fibers shows that they move from one grid axis to another largely by making right angle turns. Okay? And those turns have a radius of less than 10 microns. Most of them are 2 or 3. So it's well below any MRI resolution. So the MRI tractography is in fact in a voxel where there are a lot of turns going to show crossing fibers. Fibers coming in at one angle, leaving at another angle, but are not going to be able to resolve the fact that there were turns in there. So where do we need to go with this? Well, we need to increase the depth of the microscopic analysis, because as you saw, these 3D movies are relatively small sections in one direction, the Z direction. And so right now, one of our big challenges is in cutting, dissecting sections in the right plane so that the interesting things that we want to quantify at any point in time are largely in plane. If we can get bigger, deeper sections, we'll do it. And so many of you saw this exciting Nature paper out of Stanford called Clarity, which in fact shows a whole cleared mouse brain where all of the immunohistochemical things that I showed you could be applied. And if we could just get that to work in an equivalent size monkey brain, we could do the whole temporal lobe. or we could do the whole occipital lobe. So we're in fact starting on that immediately. My lab manager is already ordering the parts now that the paper has been published. And finally, we need to obtain statistics on the frequency and distribution of these right ankle turns. Where are they? How many of them are there? And then be able to give that to Van to compare with the signals in diffusion MRI at the loci where we will know that fiber tracts have been turning from one grid orientation into another. So with that, I'll thank you for your attention, and we've got some time for some questions.